Going to turn it on. Before we get started, I'd like to remind our students in the audience and coming in, there will be a sign-up sheet for Dr. Heidi Hanrahan's class. And we're going to put it in the front of the room. So you'll have to come up to the front after the lecture. Oh my gosh, I think this is great. I have a lot of wonderful information to share with you this evening about our illustrious faculty who are going to be presenting this evening's lectures. So with your indulgence, I'd like to get started. We are being live streamed. And for those of us who are here, I just want to say welcome to the President's Lecture Series. What a great time to turn our focus to the positive strides humanity is making in a variety of important fields. When you learn about these Nobel Prizes, you just feel inspired that there's so much creativity all around us. So a little background, in 1895, Alfred Nobel signed his last will and testament, giving the largest share of his fortune to a series of prizes in physics, chemistry, physiology or medicine, literature, and peace, called the Nobel Prizes. In 1968, Sweden's central bank established a special prize in economic sciences in memory of Alfred Nobel, known as the Sixth Nobel Prize. Tonight, we are thrilled to hear from 6 of our distinguished faculty members discussing these prizes and topics from 2024. And after the presentations, I hope you'll join us in the lobby of the Byrd Center for some delicious brownies and cookies. Now, presenting on the Nobel Prize in Physics, awarded to John J. Hopfield and Geoffrey Hinton for foundational discoveries and inventions that enable machine learning with artificial neural networks, will be Dr. Jeff Groff, Professor of Physics in the Department of Natural and Physical Sciences. Dr. Groff holds a BA in Physics from McDaniel College and an MS and PhD in Applied Science from the College of William and Mary. His expertise is quite eclectic. It spans computational modeling, biophysics, solar energy, blockchain, beekeeping, and machine learning. An award-winning educator, Dr. Groff received Shepherd University's Outstanding Teaching Award in 2017 and was named West Virginia Professor of the Year in 2019. Speaking on the Nobel Prize in Chemistry awarded to David Baker for computational protein design, together with Dimas Hassabis and John Jumper for protein structure prediction, will be Dr. Robert Warburton. Dr. Warburton is Professor of Biochemistry and Dean of the College of Science, Technology, Engineering, Mathematics, and Nursing. He completed his PhD in biochemistry at Duquesne University and then did his postdoctoral training in the laboratory of Dr. Jeffrey A. Fringer. His research interests are in proteomics, particularly in the study of point mutations and structure-function relationships. Dr. Warburton is currently engaged in some very exciting research in cancer and immunotherapy. This is all funded by the NIH in collaboration with his colleagues at WVU School of Medicine. Next, Dr. Connor Sipe, Associate Professor of Biology, will present on the prize in Physiology or Medicine, awarded to Victor Ambrose and Gary Rufkin for the discovery of microRNA and its role in post-transcriptional gene regulation. Dr. Sipe attended the College of William and Mary, earning a BS and MA in biology. Subsequently, he earned a PhD in cell biology from the University of Virginia. For his postdoctoral work, he began working with neural stem cells in the fruit fly, investigating how access to nutrients controls the development of the brain. Dr. Sipe continues his work with Shepherd undergraduate students as an investigator on a West Virginia network for functional neuroscience and transcriptomics grant, recently awarded to Shepherd and 4 other West Virginia universities by the National Science Foundation. And with us in the audience, we have a superstar, former big program director for the NSF. Thank you, Sue, for everything you've done for all of science and for Shepherd. Thank you. Speaking on the prize in literature awarded to Han Kang for her intense poetic prose that confronts historical traumas and exposes the fragility of human life will be Dr. Rachel Kranz, Associate Professor of French. Dr. Kranz joined the Modern Languages faculty at Shepherd University in 2004 after teaching French and German for several years in the St. Louis area. Dr. Kranz received her MA from the University of Munich in 1993, where she studied French, German, and Spanish. After returning to the U.S., Dr. Kranz pursued doctoral studies in French at the University of Wisconsin-Madison. Dr. Kranz's areas of specialization include 19th and 20th century French literature and culture, together with film studies. She is currently working on an interactive multimedia supplement that encourages language learning through cultural exploration. Next, Dr. Art Halslag, Associate Professor of Political Science, will present on the Nobel Peace Prize awarded to Nihon Hadonkyo for efforts to achieve a world free of nuclear weapons and for demonstrating through witness testimony that nuclear weapons must never be used again. Dr. Holzlag's expertise is in international relations, specifically in human rights, international organization, and international law. After receiving a master's in political science from Katholika Universität and a degree in human rights law from the University of Strasbourg, Dr. Holzlag worked several years for different human rights organizations. While working with Haitian refugees in Miami, he obtained a PhD in international relations from Florida International University. His research interests focus on the role of the individual in global politics. And finally, the Special Prize in Economic Sciences awarding to Dihan Asumaglu, Simon Johnson, and James A. Robinson for studies of how institutions are formed and affect prosperity will be presented by Dr. Amanda Menzik, Associate Professor of Business, Accounting, Economics, and Finance. Dr. Menzik graduated from Shepherd University in 2008. She attended West Virginia University and received her PhD in economics with a concentration in development economics. After graduating, she taught economics courses at Penn State University before returning to Shepherd University in 2018, and we're so glad you did. Her expertise is in macroeconomics, statistics, development economics, and international trade. And I would just say we are in for a very special treat this evening. Thank you so much to our speakers. Thank you.
Thank you, Mary. Is this thing on? Thank you, Mary. Thank you, everybody, for coming out tonight. I'm Jeff Groff, and I have the pleasure of telling you about the 2024 Nobel Prize in Physics, which was awarded for foundational discoveries and inventions that enable machine learning with artificial neural networks. So it was awarded to these gentlemen, John Hopfield, physicist, and what has created a little bit of controversy, Geoffrey Hinton, cognitive scientist. But the work that Geoffrey actually, the prize was shared 50-50, but if I had to say so, I think Geoffrey Hinton deserves a little more than 50% of the credit. And his work is very much based on, founded on foundational physical principles. Okay, so I want to tell you about their discoveries slash inventions, and we're going to start with John Hopfield and his invention of what's referred to as a Hopfield network. Artificial intelligence is a form of machine learning, and very much of the models that we use today are based on what's referred to as artificial neural networks. And these are inspired by, they're called neural networks because they're inspired by the way our brain is wired, where you have neurons or nodes in the model, which are connected via synapses, modeled synapses. So nodes have connections, and those connections have weights. So what Hopfield was able to do is he developed a model which is actually a model of associative memory, which is the memories we have where we recall things that are related to other things, and we remember the relationships between things. Like you might remember the relationship between a smell and a person, for example. So in his model, there are nodes that can be on or off. They're just binary nodes that can be on or off. They're connected. And the strengths of the connections are symmetric and bidirectional. And the connections are made stronger if both of the nodes are active or inactive, which is called Hebbian learning. And it's thought that that reflects the way that learning happens in our brains. But the interesting thing about his networks are you could train these networks on patterns and retrieve those patterns from incomplete or noisy inputs. So here's an example of a Hopfield network with 25 nodes. Some of them are on and some of them are off, and you have a pattern of a C, the letter C. And so when you train one of these networks with a pattern like this, and then you stimulate the network with noisy input, these are noisy Cs, the network can recall the memorized pattern by following an algorithm which is related to minimizing an energy function in physics. And you can actually memorize more than one pattern. So here is a pattern for a C, a Z, and an A. And you can then stimulate the network with these incomplete patterns and recover the memorized letters from within. So these patterns are memorized in the weights of the synaptic connections in the model. It's interesting. So here's an example where I have a model where I had it memorize C, and then I'm feeding it very, very noisy input. And actually, this one was trained to memorize C, A, and Z, and then I stimulate it with very noisy input. And sometimes it recovers C, sometimes A, and sometimes it hallucinates inverse Z. You get these spurious patterns. All right, so let me move on to Geoffrey Hinton, and his work built on Hopfield networks, and he built a model which is called the Boltzmann machine, and it's different in some very important ways. First of all, it has what are referred to as hidden layers. So, you know, I mean, this would be reminiscent of the way, like our visual cortex works. We have at least 7 layers of neurons in our visual cortex. There's a visible layer where input and output is recorded, and then you have hidden layers which are connected to the visible layer. Another big difference is in the Hopfield network, learning is deterministic. You know, there's only one solution to the synaptic weights for anything you're trying to memorize. And that doesn't really give you a good minimal energy for the synaptic weights that you're trying to minimize. So in the Boltzmann machine, learning involves minimizing a function that's like in the Hopfield network, but the learning is somewhat it has a stochastic element, a probabilistic element to it. And what this allows this model to do is to not necessarily memorize specific inputs, but to actually learn patterns and correlations in data sets. So it's actually then able to, go to the next slide. I didn't have time to code up a Boltzmann model, so I borrowed this from someone on the internet. And so instead of simply, so here are some handwritten digits, 50419, and when you stimulate a Boltzmann model with this input, what you're seeing here is a representation of what the hidden layers are learning. They are learning features in the stimulus data, like, you know, here's a vertical line, here's a hole in the middle, you know, that sort of thing. And then the model itself can then generate unique outputs which resemble the training data but aren't actually part of it. So these are all generated outputs of numerals that weren't actually in the training data. So the model learns about the patterns and the correlation in data and then can dream up new examples of it. All right, so gotta wrap up here. So why is this all important? You know, actually, the models that they've developed are foundational to AI. I would say very few applications in AI today use these foundational models directly, but the principles on which these models are based are very widespread. This idea that you are learning via the minimizing of some function or some energy function, some fitness function. Weights between connections is what encodes information and patterns in data. The Boltzmann model in particular is an example of unsupervised learning. You don't actually have to tell the model what kind of patterns are in the data. The model learns those patterns itself. This gives rise to generative capabilities. Instead of just being able to do inference, like tell whether something's a picture of a cat or a dog, you can generate new pictures of cats and dogs based on the statistics of what cats and dogs look like. I'll leave it at that. There's some other work that especially Jeffrey Hinton did, which is very important to modern AI. The use of many layers, which is called deep neural networks, and some work in optimizing efficient training algorithms for AI models. But I'll end there.
Well, good evening. Yes, it's on. There we go. I just talk quietly. So, I was lucky enough to do one of these lectures last year, and the minute I heard this was the prize, I already started working on my slides because I'm an overachiever, because I already knew some of this work and I'd actually applied it to some of the stuff I do in my classes. So I want to go back in time, not to the current group of Nobels, but this guy right here, who obviously just down the street at NIH. And in 1972, so we're looking at almost 50-some plus years ago, he made some seminal experiments that said, and led to a paradigm that said, if you know the sequence of amino acids in a protein, it will dictate the shape of that protein. So this led to everybody and their brother then working with proteins. So you want to get the amino acid sequence, you want to know the structure of the protein, and then figure out how does it do what it does. So we call this structure function analysis, right? So this is where this began. And it launched into this idea of we can get the sequence of a protein, we can find out what it does and how it does. And then maybe we can tweak it. So I've been involved where we've mutated individual amino acids in an existing protein the Nobel Prize winners were now inventing brand new proteins that have never been seen before, that are function-driven instead. So where the protein folds, you start with a random line of amino acids. They're all attached to each other. And because of their condition, they fold ultimately into their sort of the final bit, and that's the thing that does the job. But what if we mess up the sequence? What if we change the sequence? Do we lead to the same structure, or do we get a different structure? So this is the three folks, and it was Jeff mentioned the 50-50. This is a 50-25-25 in terms of how this one was split. And I knew about David Baker predominantly from work that I had done previously with students using one of his programs. That's how I knew him, right? And they always have these wonderful pictures at the Nobel site. I don't know who draws these, but I wish they wouldn't. And it sort of tells you who they are and where they were born. So this is just information straight from the Nobel site. I want to give you a different view of them. And so that's a better picture, I think, of this guy. And it tells you where he's at in terms of, so he's University of Washington. He's a Howard Hughes investigator. And the thing that was common in all of the Nobel Prize winners as they gave their lectures was how they kept giving honors and kudos to the people who came before them and the data sets that were created that they were able to work for. In other words, they could do nothing without thousands of researchers around the world helping them. And my favorite quote, he was interviewed by the Washington Post, this is it. His wife began screaming very loudly. I can just imagine, because they call him at like 3 in the morning so they can get ahead of the press. So I can just imagine his wife running around like a crazy person screaming. So here's a sequence of amino acids, and this is an energy. At the top, lots of energy and completely disordered. At the bottom, lower energy and highly stabilized. So hopefully this cartoon, oh, the cartoon didn't work, bummer. When you can do this. No, it's not. So what we have is, if I know the protein and I watch, the trick is it wants to become stable. And it does that by shedding water, and it slowly becomes this final structure. So one of the things that they did, and you can see right here, we've got University of Washington in California. This is a game called Fold It. And Fold It was designed to take advantage of people who like to play games. And so what they generated was this game called Fold It. There's a training system. There is music. You get prizes. There's little confetti explodes. Fireworks go off. And what you have is they give you a structure, and they say, fix it. Now, the people working with this are not biochemists. They could be anybody. But they do know gaming. And they know that if I move these 2 bits away, I'll get money in scoring right here. And this little horrible thing will go away, and I'll be happy. Right? That's basically it. And this is how it's being done. And so here we go. This works. Please work. Cindy, it's not working. Anyway, so you notice I've got, this is me scoring. This is 4,095. And if I can do it properly, I'll get 7,700 points. I got 7,950, by the way, just so I share that with you. But what we have are these, they gave these things, hydrophobic disasters. So all these red blobs, if you drag and push and pull, and there's some training going on, this is all computer generated. And all of you could become members of this group, by the way. Just in terms of, you just load it onto your computer, it's ready to go. But they're working with proteins that we know the sequence of. What the folks did here was they said, what if we start with what we want? How do we get to the structure, and what sequence do we need? So we'll work backwards. We'll invent proteins that don't exist. One of the things that we work with is called the protein database. And there's about 200,000 proteins now known on that base. And this is what they used as their learning system for the process. So just as a quick sidebar, so this is a open reports paper, and it was looking at various proteins. And this is what they used Fold It for. They said, we need to determine and make these evaluated folding of stuff we've never seen before. They threw it out into the world and said, tell us what you find. Play the game and tell us what the structure is. And one of the people, and there's a mess of people. You see how many people are involved in this paper? And one group I just listed as the Fold It Players Consortium, right? Because there's even more of them in there. Well, if you go to that and you break it down, this is the consortium. And if you break that down just a little bit more, you get this name suddenly appears. This is a Shepherd alumnus. This is one of my past advisees, right? And I introduced him to Fold It when he was taking my advanced protein course. Well, he played the game, and he got a publication out of it, which is kind of cool. He's actually an author on this paper. Now, you may say, oh, that's cool, but Mike Faggiola is one of our 4 under 40. It's this guy right here, right? In terms of, and I had the pride to say, I know all of these students. He was students of mine. And so, for example, Mr. Goldman, right, his wife just delivered twins, so he doubled his family in one event. Congratulations. Mike Faggiola, this is him, is a graduate now, PhD of St. John's in New York, and currently is the deputy toxicologist for the state of Maryland. That's what he does for a living. This is it. And so I'm going to take all the credit for that. That was all my stuff. So that was the first one. The second group is this guy right here. And this guy was an overachiever from a very young age. He was introduced to chess at the age of 5, and by 13 he was a master. And then he started playing with games that played chess and figured out how could he make them better so they could beat him. And eventually he sort of went up. And one of the things, he got bored with chess and said, there must be a tougher game for me to play. And the game was Go. And so this is basically black and white tiles, and there's thousands upon millions of variations on a theme. And he developed computer programming to play Go. In fact, they beat a master of Go from South Korea in terms of that process. But they then said, well, can we apply it to an even bigger problem? The problem was proteins. And so they worked backwards, right? And in his code is this one right here. It's like, you have to ask the question. So, example, we have a problem with plastics in the environment. We need to get rid of the plastics in the environment. We know what the structure of the plastics are. Let's invent a protein, an enzyme, that will destroy the plastics. So what they do is they take the shape of the plastic and they just tell the computer, based on gaming, make a protein that will bind to this thing really well and kill it. That's how you invent, for example, protein vaccines. That's how you invent drug systems. So the import of this process is they're inventing proteins that have never existed. They're all computer generated. But then what they do is they then synthesize those proteins, and lo and behold, they do exactly what they were designed to do. Now remember, there was 200-some thousand that they're playing with? This guy, John Jumper, in terms of the process, they went to one company, Deep Thought. Deep Thought was eventually sold to Google, right? So it's over in there. And they invented a program called Alpha. Alpha 1, then Alpha Plus, and then Alpha Plus 1. And what they did, there's a competition each year to help design new experiments and new pieces. And for years and years, over a decade, the grade on the exam was 60%. That was as best as they could go. When they went to AlphaFold 2, it got to 90%. We went from 60% to 90% prediction, and then they sold the company for $1 billion+. So it was kind of nice for them in terms of the process. But what happened was, it was kind of interesting. They were surprised that they would even be recognized. And when you watch this guy's Nobel lecture, for example, I have a bottle of water, he almost choked on his water when he was doing his presentation because he said he was so nervous. The guy just won a Nobel Prize. What's he got to be nervous about, right? And so this is the alpha system that they work with, and they generate this program. This protein right here is interacting with that protein. This has never been seen. This has never been seen. It was designed by computer to fulfill a particular job, and has subsequently been synthesized by the process, and it does the job interesting. They again used the protein database as their baseline. That was 100 and some thousand proteins. They then have applied it to everything they can lay their hands on, and if you look at this number, they're now looking at 200 million proteins that they've looked at. So exponentially, it's a lot bigger, and we can predict lots of different things. So I'll give you an example of SARS and COVID. It mutates. Knowing the mutation, you can use this system to design the next vaccine system from it, because the learned machine doesn't care what happens. We have avian flu. We can devise vaccines for avian flu because all we have to do is get the model and then let the computers design the protein that's going to interact with it and then send it off for generation as the new vaccine. So this is sort of my last slide of the piece. And it was coming back to Jeff's commentary about AI and machine learning. This is what this is. This is applied AI that we're looking at. And one of the things that they said is this dual purpose pieces. And one of the things is this part. The AI carries both risks, and those have to be aware of. And so when I was making this slideshow, something popped up. January 27th, suddenly that appeared. This is an AI programming out of the People's Republic of China, effectively. And did it have any effect? Yeah, This is the stock market on the day that it was released. And you suddenly go from there down to here. And these are companies like Oracle and NVIDIA who are associated with AI programming and machining. So we're looking at it. I give you good examples. Destroy plastic toxins, generate vaccines, generate nutritional materials, generate... AI can go this way as well. So we have to be careful. And scientists, when we started doing genetic engineering, met in a Silomar in California and said, we are the experts and we will control how this is done. This is something we need to look at as well, because this can be used, because I can also design bioweapons using this methodology. I've given you the good side. Do I want to take you to the dark side? And that's my talk. Thank you very much. Come on, Ethel.
All right, tough act to follow there. I think if we had been thinking, maybe we should have put my talk before his, because I'm going to talk about the biology that comes before the proteins. So we're going to kind of step back in the flow of things just a little bit. And so I'm Connor Sipe, talking about the prize in physiology or medicine, and this is for the discovery of a new kind of nucleic acid in cells that's known as microRNA. And so you may or may not know this, but the human body has around 32 trillion cells in it, average human body. And the amazing thing is, despite the fact that we have all different kinds of cells, they all have the same DNA inside them. Okay, so the genome of every cell is exactly identical with very few exceptions throughout the body. And so this has been known for a long time, but a fundamental question in biology is, well, we know we've got different types of cells in our body, right, that have very different structures and functions. So on the left, we've got a neuron that has a cell body there in the middle. It's sending out branches in every different direction so that it can communicate with other cells. On the right, we've got this hexagonal, flat, plate-like skin cell. Even though these two cells have very different structures and functions, they have the same DNA. So how is it that our body can generate these different cells, right? If they all have the same blueprint inside them, how does that work? Okay. And so that's what this year's prize is really all about. It's elucidating a little bit about how that works. So when you're thinking about different cell types inside the body, so something like a skin cell or a blood cell or a neuron, they have different structures and functions. Okay? And so what makes cell types different from each other is the set of proteins that are inside the cells. Just what Bob was talking about. Proteins are the molecules inside cells that actually do things. So they make chemical reactions happen, they move things around, they carry things across the cell, they do all kinds of things. And so depending on what type of cell you are, you will make a certain set of proteins, and those proteins allow the cell to carry out its job. Okay, so a neuron in the brain is going to express or make proteins that allow it to communicate with electrical impulses. The skin cell would create proteins that are very structurally sound and protective. Okay. And so the way this works is the cells all have the same library of genes inside them. Okay, so each individual gene encodes one protein. Okay? So all the cells have the same library, but then the different types of cells are going to choose which genes out of the library to actually make into proteins. Okay, so each cell type will end up making kind of a unique mix of proteins. Okay, and that is going to give them their specific characteristics. Okay. Now, the way this works is there's a specific flow of information inside the cell. Okay, so we're all very familiar with DNA. All cells have DNA in their center. The point of DNA is to store genetic information. Okay, so that genetic information encodes What it actually is doing is it's instructions for making proteins. And the cell has a very specific process that it goes through to take the information stored in DNA and use that information to make a protein. Okay? And that process is referred to as gene expression. Okay? So different cell types, the neuron and the skin cell, express different genes. Okay? Now, it was long thought that the important process that really determines what proteins a cell will make occurs at this step known as transcription. Okay? When a protein is made, the information in DNA resides in the nucleus, and it stays in the nucleus over the long term, but it's when a protein needs to be made, that information is copied into a molecule called mRNA, that's messenger RNA, and that will make its way out of the nucleus into the cytoplasm, and there that information is used to make a protein. Okay? And so it was long thought that this step right here, a cell will decide which books in the library, if you will, which genes in the library of information will be made into RNA. That's going to be the critical step in determining what proteins are eventually made inside a cell. Okay? And it turns out that this year's Nobel Prize winners kind of turned this paradigm on its head. This idea is so famous, it's actually called the central dogma of molecular biology. But this year's Nobel Prize winning work by Victor Ambros and Gary Ruvkin kind of turned this on their head when they discovered another kind of molecule that's very important in determining what genes are actually made into proteins. And it's kind of interesting that I got asked to do this because I have a very tangential connection, academic connection with these folks. They both worked in the Horvitz lab at MIT, which is where my PhD advisor worked as well. So I'm kind of like somewhat connected to them. So that's kind of cool. So nowadays, when they did this work at MIT, but nowadays, Dr. Ambrose here is at the University of Massachusetts Medical Center, and then Gary Ruvkin is at Harvard Medical School. And these folks were working back in the early 90s, so we got to go back a little bit for this prize. These folks were working on this organism right here, which is called C. elegans. It's a tiny little nematode worm that lives in the soil, out everywhere. Essentially, there's all kinds of them. We never see them because they're so small. But they're very good for working in biology because a lot is known about their genetics. They can be manipulated very easily. And so our Nobel Prize winners were working in trying to understand the genes of this worm. Okay? And what happened was Dr. Ambrose he discovered a gene in the worm called Lin4, and this gene was very, very small. Okay? It was only 22 nucleotides long. A normal gene is several hundred to 1,000 or more nucleotides long. So this was a very short gene. And when they looked at it, they saw that there's no way it could actually make a protein. Okay? And so it was very curious that the worms would have this because it was thought at the time, well, the point of genes is to make proteins. It turns out that they got to talking, and over a period of time, they worked out that this particular gene, Lin4, what it does is it actually binds to the mRNA of another gene called Lin14. Okay, and Lin14 is a gene that makes a protein. Okay, what it does isn't really that important for this talk. But Lin4 is made, okay, this short little piece of nucleic acid is made, it goes out into the cytoplasm, and it finds the Lin14 mRNA. And when it binds to it, it prevents this mRNA from being made into the Lin14 protein. Okay? So when the worm makes Lin4, it shuts down the ability of the worm to make this other gene. Okay? And so there's no translation, and so there's no Lin-14 protein made. Okay? And at the time, this was, it wasn't received with widespread acclaim because people were worried it was kind of a worm-specific thing. Maybe, it's not important. But it turns out that since this discovery, it took them about a decade to discover another RNA that does this. But since then, they've been discovered all over the place. And in fact, this Lin-14 that was first identified is a new type of RNA that had never been seen before. It's called microRNA because it's so short. And it represents an entirely new way for a cell to choose which genes are made into proteins. Okay. So returning to our central dogma here, yes, choosing which genes to express is important at this stage. Choosing which genes from the library to make into proteins is important. But our Nobel Prize winners discovered this intermediate or new layer of control over gene expression that uses these small microRNAs to bind to the messenger RNA and prevent them from being translated into proteins. Okay, and so since that time, microarrays have been discovered all over the place. All plants, animals, and fungi use these regularly. So the human genome at this point there have been over 2,000 discovered, and it turns out that those 2,000 microRNAs regulate or control about 60% of the genes that are expressed in human cells. So they're very, very important for essentially anything a cell does. Okay, so development, just the day-to-day function of our cells, and of course disease states. And I'm just going to wrap it up by showing you an example of why this is so important. It's now appreciated that cancer cells use microRNAs for nefarious purposes all the time. Okay, so I think we all know cancer cells divide uncontrollably. They want to spread out through the body. Cancer cells are very, very good at subverting the normal systems that are in cells and hijacking them for this purpose so they can grow faster and spread. And this new layer is no exception. Cancer cells, essentially any cancer type you can think of, have been found to manipulate the expression of these microRNAs so that they have tighter control over the cell physiology so that they can warp it to do what they want to do. So this is with this discovery that cancers use this all the time, it's opened up new areas for both detection of cancer as well as treatments of cancer. So if cancer is using this system to control cells inappropriately, let's take this back from cancer and shut down the systems they're using to hijack the cells. So I hope that's a good overview for you. Thank you very much.
Is this working? Okay. Help. Okay. All right, I'll try it this way. Can you guys hear me? Yeah. All right, so I need some notes because I have some quotes and I can't remember them all. Okay, so I'd like to talk to you tonight about the work of Han Gang, the South Korean author awarded the Nobel Prize for Literature in 2024. Quote for her: "Intense poetic prose that confronts historical traumas and exposes the fragility of human life." End quote. Her selection is noteworthy for two reasons. She is the first writer from South Korea to win the prize, and she is a woman. Historically, women have been underrepresented in this category, comprising only 15% of recipients. To date, Han has published 8 novels, several novellas, as well as collections of essays and short stories. I'm going to recommend 3 novels, in case you're interested in reading something by her, that have been translated into English. Although very different in their subject matter, all 3 are relentlessly political in their exploration of what it means to be human. The first is Greek Lessons, translated in 2023. It's a love story of sorts that focuses on two individuals who are losing the most vital links that connect them to the outside world, namely language and vision. As a review on the Nobel Prize site explains, this novel, quote, traces their attempt to find a common ground in their shared bereavement, end quote. The second work is Human Acts, translated in 2017. Here, Han explores the repercussions of the Gwangju uprising that took place in May 1980. After the military coup by Chen Dong-hwan, university students took to the street to champion democracy, only to be massacred by government troops. The third is entitled The Vegetarian, translated in 2016. This is perhaps Han's best-known work, as it was the first to be translated into English, and it is this text that will be our focus tonight. Okay, so this novel has a three-part structure similar to a triptych or the movements of a symphony. As far as the subject matter goes, it's easily summarized. After 5 years of uneventful marriage, the protagonist, Young-hae, unexpectedly announces that she will no longer consume any animal products, a decision that shocks her family and stuns her social group, since it goes against prevailing norms in South Korea. Their astonishment and incomprehension mount as Young-hae seems to descend into madness, refusing to eat entirely in an effort to cast off her human form and become a tree. Yes. Okay, what is harder to convey is how this story is told. Each section of the novel has its own tone, a color, and a flavor that sets it apart from the others. Each section also has a different narrator who highlights the impact of Young-hae's decision. The first section is told from the point of view of her husband, the second from that of her brother-in-law, and the third from that of her older sister, In-hae. You may wonder why we almost never have the protagonist's point of view. In a 2023 interview, Han explained, quote, "I wanted to show through the narration of the other characters how Young-hae is observed, hated, misunderstood, pitied, and objectified. I imagined the moments when the readers piece together her truth as it emerges from these misunderstandings," end quote. So, Section 1. Young-hae's decision constitutes, quote, "an appalling change," end quote, for her husband, similar to what happens to Gregor Samsa in Kafka's Metamorphosis. And although she wasn't directly inspired by Kafka, her work is often referred to as surreal and Kafka-esque. So just you know what you're getting into. The husband finds her decision to forego meat incomprehensible because until that point she had been, quote, "completely unremarkable in every way," end quote. During their marriage, she had always had a passive personality, had been demure and restrained as women in South Korea were traditionally expected to be. Now, however, she is cool and self-possessed, stating categorically, "I won't eat that," rather than apologizing for what is seen as crazy behavior. In addition, she refuses to provide an acceptable reason for her decision, for example, a medical condition or concern for the environment, citing instead her dreams as the catalyst. Passages in italics give us a sense of these nightmares and justify Young-hae's desire to purge herself of the violence innate in all human beings. Quote, "They come to me now more times than I can count. Dreams overlaid with dreams, a palimpsest of horror. Violent acts perpetrated by night. A hazy feeling I can't pin down, but remembered as blood-chillingly definite. Intolerable loathing so long suppressed. Loathing I've always tried to mask with affection, but now the mask is coming off," end quote. Neither Young-hae's husband nor her family are interested in the reasons for her decision, which they find unreasonable. Instead, they urge her to resume eating meat, declaring that, quote, "vegetarianism goes against human nature," end quote, and that, quote, "a balanced diet goes hand in hand with a balanced mind." When cajoling and threats have no effect, Young-hae's father has her held down so he can make her obey him and eat meat. She, of course, resists his violence, then slits her wrist in an effort to reclaim autonomy over her body, creating a pool of red that recalls the slaughtered animals she sees in her dreams. The first section is thus marked by resistance, violence, and blood. In the second section, Young-hae becomes the vessel for an artist's dream. This section takes place 2 years after the first. Young-hae is now living alone after her husband filed for divorce, unable to deal with the shame she had brought upon him and her family. No longer tied down by social expectations, she is open to life, embracing energy and desire. This non-conventional attitude fascinates her artistic brother-in-law, who is Inhe's husband, as does the knowledge of a small blue birthmark, a so-called Mongolian mark, that she has on her buttocks. In a flash of inspiration, the brother-in-law sees the birthmark morph into a blue flower, its petals opening outward. From there, he pictures her entire body painted with brilliant blossoms and envisions her copulating with a similarly painted man. However, as Hahn makes clear, this is not a sexual obsession, but rather an artistic vision. Quote, covered with flowers and leaves and twisting green stems, those bodies were so altered it was though they no longer belonged to human beings. The writhing movements of those bodies made it seem as though they were trying to shuck off the human, end quote. When approached by her brother-in-law, Young-hae agrees to become his canvas. Her calm acceptance of this process makes her seem something sacred, a mysterious being with qualities both animal and vegetal. Quote, a thrilling energy seemed to flow out quietly from some unknowable place inside his body and collect on the tip of his brush. Orange daylilies bloomed on her concave stomach, and golden petals were scattered pell-mell over her thighs, end quote. And once he has filmed her flower-covered body, he gets a fellow artist to paint him so that he and Young-hae can conjoin their bodies and thus make his vision a reality. This section is thus marked by vibrant colors, floral motifs, openness, and desire. This positive tone ends abruptly, however, as Inhe discovers her husband's infidelity and has both him and her sister committed to a mental institution. Yeah. And I'm going through this because I want to give you a feeling for what the work is like and maybe inspire you to read it for yourself. So in the third and final section entitled Flaming Trees, Young-hae continues her journey beyond the human, trying to merge with the earth and become a tree. And this is where it gets particularly surreal, except that the setting is entirely realistic. And so the line between madness and sanity is very thin here. So this takes place 1 year after the previous and is told from Inhe, the sister's point of view. Young-hae is near the point of death, having decided 3 months earlier to stop eating altogether. At that time, she had escaped from the asylum to go to the forest where she had been found, quote, stock still and soaked with rain as if she herself were one of the glistening trees, end quote. She had begun the process of becoming one with nature. Consequently, there is nothing depressing about Young-hae's hunger strike, because she sees it as the last phase of the transformation she began by becoming a vegetarian. Quote, her eyes shone. A mysterious smile played on her face. I'm not an animal anymore, sister, she said. I don't need to eat, not now. I can live without it. All I need is sunlight, end quote. Yet even this joy is denied Young-hae as the asylum doctors insist on keeping her alive. In a scene that echoes the force feeding in the first section, Young-hae is held down while the doctors try in vain to insert a feeding tube through her nasal passage. Eventually, Inhe intervenes, telling her sister, it's your body. You can treat it however you please. The only area in which you're free to do as you like. It's interesting to note how Inhe's feelings for her sister include not only pity and despair, but envy as well. It is true that Young-hae is confined to a mental hospital and has wasted away to mere bones, but she is living on her own terms, which makes Inhe realize that she herself has never truly lived, merely endured. She has always done things the right way, caused no harm. Young-hae, however, has shucked off social constraints and left her behind, still a prisoner. Inhe is, quote, unable to forgive her sister for soaring alone over a boundary she herself could never bring herself to cross. Before Young-hae had broken those bars, she'd never even known they were there, end quote. This section is thus marked by self-reflection and the specter of death. Yet it ends on an optimistic note with the forest brilliantly aglow after a summer storm. Quote, in the waning afternoon light, the rain on the leaves glitters intensely, kindling a green fire, end quote. I read this image as emblematic of the strength and the endurance of nature, highlighting the power that comes from the immutable union of sun, water, air, and earth. This silent power is what Young-hae longs for. This is the goal of her resistance and her personal transformation. So I hope this brief discussion of the vegetarian has kindled a desire for you to read it for yourself, since the text is far richer and more complex than I could convey here.
Hello, everybody. Thank you, Rachel. Like Rachel, I also have some notes because it's not easy to remember everything. But yeah, so I'm talking about the Nobel Prize for Peace, which was awarded to Nihon Hidankyo, which basically means the organization of sufferers of atomic and hydrogen bombs. This is a Japanese organization that was established in 1956. And I actually have a personal connection to this to some extent because my father was a survivor of the Nagasaki atomic bomb. If he were still alive, he would be 102 by now. But he would probably be a member of this organization or somebody that they would be advocating for. The objective of the organization is to basically try to eliminate all nuclear weapons and to advocate for the rights and the treatment of the survivors of atomic and hydrogen bombs, as well as, so not just Nagasaki and Hiroshima, but also the tests that were done with atomic bombs. And actually, their first appearance on the international scene was at the World Conference on atomic and hydrogen bombs, which was 2 years after the nuclear test by the United States near the Marshall Islands, in which 23 also Japanese fishermen were poisoned with radiation, and many people in the Marshall Islands as well got infected with the radiation. So, and this is a problem that goes much further than the 2 atomic bombs. But just to give you a little bit of a background, of course, Hiroshima and Nagasaki, I think everybody at least has heard of this. August 6th and August 9th, 1945. In Hiroshima, 70,000 people died at the moment of impact. 140,000 people died in the between August and December 31st. And then, how many people were affected by radiation and other effects of atomic weapons is unsurmountable. In principle, this means that 55% of the population of Hiroshima was eliminated. Nagasaki, we're talking a little smaller number. We're talking about, where did I have it? Sorry, 40,000 people killed immediately and 70,000 in the aftermath. In Nagasaki, it meant that 40% of the city, the infrastructure of the city was leveled. So what we're talking about here is immense destruction. And if you think about this, over the years, since 1945, the Nobel Peace Prize Committee has awarded 6 different prizes before this one to organizations or people advocating the non-proliferation of nuclear weapons. And that tells you, in my mind, at least some of the importance of this topic. The organization itself had been put up for this prize on several occasions, and finally in 2024 they received the prize. This is something long time coming. The main objectives of this organization As I said, the prevention of nuclear war and the elimination of nuclear weapons, including the signing of an international agreement for a total ban and the elimination of nuclear weapons. Secondly, and this is of course why it is an organization of those who have been affected by nuclear weapons, state compensation for the damages done by atomic and hydrogen bombs, and the state responsible accepting that responsibility and creating laws and procedures to help those who suffer from the aftermath. And so that's the last part, improvement of the current policy and measures. And the assistance of the hibakusha, which are the survivors of nuclear weapons. Yeah? What does the organization do? Well, it is a social movement. So what does it do at protests? But their special power is that they are, of course, an organization of survivors. Now, as you, of course, noticed, as I said, my father would be 102 if he were still alive. These people are getting older and older, of course. But much of what they have been trying to do is to use first-hand accounts to tell the story of what the effects of nuclear weapons are and bring those to international conferences, to politicians, to help make people aware of the effects of nuclear weapons. Secondly, of course, they protest, they do letter writing, etc., all the things that social movements do. If you have any idea about the work of Amnesty International, for instance, it is not that different in that sense. But they, of course, because they are have first-hand accounts, have a special role to play in this whole situation. It is especially important still today if we look at the Atomic Scientists Organization. They have something that they call the doomsday clock. Doomsday is, of course, when the next nuclear weapon will explode. Well, that clock for 2025 is at 89 seconds to midnight. Because nuclear proliferation is expanding, and expanding rather rapidly, I would argue. As I said, Russia has basically suspended its compliance with the START Treaty, which is a major nuclear weapons non-proliferation treaty. It has withdrawn its ratification of the Comprehensive Nuclear Test Ban Treaty, which of course is the treaty that that stops the nuclear testing, and at a certain point in the 1990s basically stopped every country from testing nuclear weapons. But that is not the case anymore. Of course, as some of you know, North Korea has tested lately. And North Korea, of course, also has tested ICBM, intercontinental ballistic missiles, which would allow to carry a payload across continents, which, of course, the ICBM treaties that were created between the Soviet Union and the United States in the 1980s were a major reason why the Cold War went into a freeze. And so the fact that North Korea is using those again, is testing those again, is a major problem. So did China. China has been also been testing intercontinental ballistic missiles, and has expanded its warheads. And by the way, so is the United States. The United States is upgrading its nuclear arsenal. is spending more money, and this started already under the Biden administration, actually already under the Obama administration. Yeah. The United States, China, Russia, the big powers in this world seem to have kind of gone away from non-proliferation. Then, of course, we all know that there, or at least I hope, that there was an agreement with Iran during the Obama administration that then was cancelled by the Trump administration. Since that cancellation, Iran has been upgrading its nuclear arsenal and is now very, very close, basically weeks away, of creating its first nuclear weapon, which of course in the current situation in the Middle East might be very problematic, since the whole order in the Middle East has dramatically changed. And Iran might feel itself to be pushed into a corner. And of course, being pushed into a corner might lead to drastic measures. And so if that is the case, the most drastic measure is a nuclear weapon. I see this personally, and that's my personal opinion, as a major threat in this world at the moment. And I would actually say we might be closer than 89 seconds from midnight.
Well, good evening, and thank you for being here. It's a pleasure to get to talk a little bit about some of the work that actually kind of I first became acquainted with when I was an undergrad here at Shepherd. So tonight I'm going to talk about some of the work in economic development by Darren Asamoglu, Simon Johnson, and James A. Robinson. And what first inspired them is something that a lot of us look at, is when we see a map of the world today, especially if we can color code it and think about what the GDP per capita looks like around the world. We see some very stark differences, and especially in a world that has become increasingly globalized and interconnected, and it seems like there's a lot of sharing that could potentially go on. It's amazing in a lot of ways that we still have such stark differences around the world. And so one of the things that has been looked at for quite some time is a couple of patterns that seem to persist. And if we stay on this map here, but just make a little tweak here, just add in 2 lines. This is the Tropic of Cancer and the Tropic of Capricorn. And one of the things that has been noted, that with a couple of exceptions and also the tip of Australia here, for the most part, this represents the poorest part of the world, okay? And so this has been very much linked to the fact that it is located very close to the equator, which means that it's very extreme temperatures, often erratic climate, could be hurricanes, typhoons. Also, incidence of tropical disease is going to be most prominent in this part of the world as well. So these types of things have been looked at for quite some time as some of the things that could be keeping back this part of the world from being able to advance economically, okay? So from this perspective, in a lot of ways, it's almost like, again, where you are on the map is very deterministic in terms of how successful you're going to be able to be as a nation, okay? And there was an article that I remember reading here at Shepherd when I came here that was entitled that, Prisoners of Geography. And it was this idea that, again, where you were on the map was going to basically tell you how far you could get, okay? So one of the things that our Nobel Prize winners challenged is this idea of geography and this hypothesis. And specifically, they were looking at this idea that if a country was again, we're not going to be able to change the geography of a country. So if a country was at one point in time wealthy, then it should always be wealthy because it's, again, at that same spot on the map, okay? By the same token, if a country was a poorer country, then we would imagine it would always remain poor because, again, it's on its spot on the map, okay? So they started to look at data historically, and they saw that this was not the case. In fact, they were able to observe what they referred to as a reversal of fortune, okay? And so what this map is looking at here is if we look at the vertical axis here, going up and down, we're looking at GDP per capita. Now, this is from 1995, but we're going to consider that to be current GDP per capita, okay? Against urbanization in 1500, okay? We'll talk about the importance of that date in a second. But the idea here is urbanization is supposed to be a proxy for income per capita because we weren't getting measures on that kind of information at that time, okay? So the idea here is that we would expect to see a positive relationship, an upward sloping curve if we were to draw a line of best fit going through the data, okay? A country that is poor in 1500 would also be poor in 1995, and a country that was wealthy in 1500 would also be wealthy, okay? But we actually see as the opposite, and this represents this reversal of fortune, okay? So we have, again, countries that, up here we can see USA, Canada, and Australia right off the bat, which we know are wealthier countries today. If we think back to the time of 1500, these were very sparsely populated areas of the world, okay? And they experienced this reversal of fortune, okay? And so we're going to look at some other civilizations that were much more developed, if we think about, again, relative to the rest of the world in 1500, and that included civilizations like the Mughals in India, the Incas in South America, and the Aztecs in Mexico, had advanced civilizations. And at this time, again, if we compared to other parts of the world, which were much more sparsely populated, North America, Australia, New Zealand would be examples of parts of the world that were much less developed, okay? Okay, and so again, what the previous graph was showing us is that over time, these civilizations would actually switch in terms of they would become less economic developed over time, and these would take the lead. So again, this flies in the face of what was had been thought originally in terms of the geography hypothesis about where you were on the map was all that mattered. Okay, so there was something else that was driving this. Okay, so before we go any further, though, I just want to introduce the idea of institutions because that's very important for what their work is very much centered on. So it's a very broad category. It includes customs, rules, social norms, okay, and so they can be both formal or informal. Formal institutions can include law code, the form of government, if there's a written out constitution, okay, all would be considered formal institutions. But then there's also informal institutions. And this can just be, like I said, social norms, customs. If you shake someone's hand, does it mean that you're going to make good on a promise? Again, tipping, local bartering practices. So it's a very large category. But what we're going to focus on is a couple of different types of institutions that Acemoglu, Johnson, and Robinson are really going to single out as what were some very important determining factors in the way things changed. Okay, so we want to go back to that date of 1500. Why 1500? And at that time in the world, it kind of marked a really big change in terms of the way the world powers were going to start to be distributed. This was around the age of exploration. Europe was going out and getting ready to colonize the world and really put its influence all around in a way that we hadn't seen before. Okay, and so the work that the Nobel Prize winners went into was starting to look at where did these European colonizers settle and what did they do when they got there, okay? So there's two different paths that we can look at in terms of the colonial strategies that were observed. And it had to do with when they went to a new location, what was that location like in terms of, was it a nice place for people to want to settle down, okay? And so we're looking here at what the mortality rates were of the settlers, okay? And so this would be determined based off of if there was any kind of disease problems in that particular area, if it was a nice, desirable climate to be within. Okay, so if it was a nice place to be in, then this seemed like a good place for some of the colonists or the colonizers to actually settle down. Okay, and so the idea is that they could actually move over here, bring their families, and start their own colony. Okay, so if you're going to start your own colony, you want to bring with you the things that are going to make life nice. Okay, and so this is where, again, the Nobel prize winners started to look at this idea of what kinds of institutions do they bring with them. And they named them inclusive institutions. And we'll talk more about what kinds of things that represented in a second. Okay? So what we're going to see is that this low settler mortality ultimately became related to high current income per capita countries that we're going to be looking at today. Okay, so starting to explain why we're seeing the flip or the relationship, how things started to change. Okay? So the other path would be if Europeans went to a different part of the world, and when they got there, maybe disease was a really big problem. It did not seem like a nice place, again, to bring families and to want to start a new colony. But at the same time, once you land there, it's not, they still want to be able to benefit from, again, this new opportunity somewhere around the world. Okay, so instead of living there, they would find a way to still, again, benefit from it. And so they created non-settler colonies, and with that, set up extractive institutions. And the idea was maybe they could get from it different raw materials, whether it was mining gold, silver, or, again, whatever raw materials were available from these colonies, okay? And so, as you can imagine, if you're going to be getting things out of a colony, you're going to set up a very different set of institutions in terms of what you need to do in order to extract from a colony, okay? And so, again, Acemoglu, Johnson, and Robinson named these extractive institutions. What we see is that these types of colonies, again, that were non-settler colonies, they are associated with the low current income per capita that we see today. Okay, so a little bit more on extractive institutions versus inclusive institutions. Again, extractive institutions were for the purpose of resource extraction, okay? And so some of the types of institutions that would have been set up would be, again, to try to benefit the home country without having to fully be there, okay? So you would have to kind of set up more of a ruling elite that could take care of, again, helping to make sure things were coming out as planned, okay? And this was done at the expense of, again, indigenous populations. So they would have to deal with institutions that would include taxation and labor exploitation. Okay, so again, things that would help to prop up this extractive type of colony that they're creating. Okay. On the other hand, if Europeans were moving to a new colony, of course, the purpose would be to create a home for themselves, and they would bring with them these inclusive institutions. And so some of the ones that they would bring would be property rights, rule of law, democratic governance, all things that have been associated with increased levels of income, okay? Property rights really looked at as very important in terms of people wanting to really invest the time into, let's say if you're a farmer, if you have land and you know that nobody can take that land away from you, you're willing to work harder to reap the gains from that land, okay? If you think the government can take that away from you or your property rights won't be respected, you're going to have a lot less incentive to want to work the land, okay? So just a basic example of how this really drives economic growth when people know they have those kinds of protection. Okay, and so it's very clear when we look at a graph here, again, where Europeans settled based off of this new way of looking at things. So again, we have GDP per capita in current times on the vertical axis. And over here, we have the settler mortality rates. Okay, so higher levels of GDP per capita today are associated with the areas that had lower settler mortality rates at the time that Europeans were first going out. Okay, so it's clear that, again, this falls in line with this pattern of, again, Europeans wanting to settle in places, again, that have lower levels of mortality rates when they arrived. Okay, we can also look at this in terms of this idea of a reversal of fortune through this cartoon, and it looks at the idea of how there were these civilizations, that were more advanced originally, okay, if we compare them to, again, the more sparsely populated areas of the world, okay, at the time of 1500 and prior to that. We see the arrival of Europeans as they started to colonize the world, again, around the 1500 mark. But we see it doesn't change right away. The reversal of fortune actually really, according to the work that they did, started to take hold during the time of the Industrial Revolution. And this is evidence that, again, this idea that some of these inclusive institutions that had been put in place really started to allow the economies to benefit at this time when there was this new technology, new ideas, things taking off in a way that the countries that did not have these inclusive institutions could not have the same benefits. Okay, and so we see instead that the countries that were formerly less developed, not as wealthy, they actually come onto this new trajectory altogether, and they're able to surpass the countries that had formerly been the leaders of the world, if you will. So to summarize, there's some very important contributions from the work that these economists have done. First of all, being able to break free of this idea that geography is going to rule everything when it comes to your success as a nation, I think is a really important concept and idea, and to literally show that the same spot on the map has experienced success in the past, and we would imagine, again, has the opportunity to potentially have success again in the future with the right institutions in place. The other thing that it does, though, is it also demonstrates the lasting effects of institutions over time. Okay, so we're past this colonial time, okay, and we've seen colonies have gained their independence, and now they're, again, their own countries, but there still is this persistence. There was this idea of setting up a ruling class in a lot of different areas in the world, and even, again, as these colonies gain their independence, we still see that some of those institutions, the extractive institutions that were originally put in place, have continued to kind of remain in there and haven't really fully left. Okay, so it shows that institutions are very long-lasting, very deep-rooted, and they take a long time to really fully, again, get rid of them if you're trying to get rid of it, what would be considered a bad institution. And lastly, of course, we can see that economic progress is very much driven by these inclusive institutions, the opportunities that they give all citizens to be able to prosper economically. Thank you very much.
Thank you so much to all of our faculty. We can take a few questions. Does anyone have any questions for any of our presenters tonight?
Most of this is beyond my reach, but I am always struck by this gathering. And I'm curious always about whether or not it's a real pleasure to be with faculty. I'm always wondering if you all know each other and if you have opportunities to sort of be outside of your individual silos and talk together about things that may not be in your particular area either. How much time is there within this busy life of all the work that's involved to get acquainted, to like each other or dislike each other?
Well, it depends. I think that I personally know the people within my academic program very well, and within my college fairly well. But contact with faculty outside of my discipline in college is pretty much limited to committee work. And that doesn't really lend itself to getting to know each other very well. But there's exceptions, you know. I don't know. I have made friends outside of the department, but I feel like we could do better at being more widely collegial in that way. But maybe some of these other professors feel differently.
Sorry, completely different topic. During the talk on the Peace Prize, there was the idea of state compensation that was mentioned. And I guess the question that came up to my mind a few different times was, when we talked about state actors, were we discussing, as far as compensations, were we talking about the Japanese government, or in the case of what happened there, the United States government?
So the idea is very clearly the Japanese government. The way that the organization states this is that Japan started the war. And this is already a very controversial statement within Japan. As you might know, Japan has still this day not apologized for the war. So the organization, as a Japanese organization, stating that Japan is the reason for the war, and therefore is also the reason why the atomic bomb was used. So therefore, it is the Japanese government who would have to compensate. However, if we are talking about testing, then it is, of course, yeah. So if we're talking about the test by the United States in the Marshall Islands, then it would be the United States that would have to compensate.
Any other questions? Take one last one.
Actually, I'm not sure what my... Is this on? I've been thinking, kind of putting some of the presentations together. Artificial intelligence and nuclear weapons. What does that lead us to? There is a book I read recently called Nuclear War, an Assessment, and it's probably one of the most depressing books you can read, because it takes a hypothetical nuclear attack by Korea on the United States, which eventually leads to an overall nuclear war, which will probably eventually lead to the destruction of the Earth, as we currently know it. So, and I'm not very optimistic that is not going to happen at some point. And Iran is a very good example of that. How do we stop them from getting a nuclear weapon? How do we stop them from attacking Israel? So.
In my current feeling, but that's more dealing with students, AI is a nuclear bomb. But just because of how students use AI and how it undermines, in my mind, at least for the way that they use ChatGPT, critical thinking and analysis. And as such, I think that's a major problem. And I think that problem is very similar to the problem of nuclear weapons, in that this is new technology in the sense of even the technology that Alfred Nobel invented, which has consequences outside of the original intent. And these consequences need to be guided, guarded, whatever you want to say, yeah, however you want to call that. And of course we have a history during the Cold War of somewhat responsible actors on the world stage. And I'm on purpose saying somewhat responsible. We are, of course, at a point in time where the memory of nuclear disaster is second-hand. Yeah, as I said, I mean, there's a reason why I said my father is a survivor and he would be 102 years old now. Yeah, that kind of a story I only know because of him telling. And so the effects of nuclear weapons are not that clear to the current leadership. Yeah, I'm 62 years old. Yeah, most leaders are my age or younger, except in the U.S. Yeah, but you know, and then we can think about responsibility and just the knowledge of what the effect might be. And then, of course, my point about Iran is that the use of weapons in general, I mean, how does a bar fight start? At the moment that a person feels they have no other way than protecting themselves than with physical violence. Yeah, if you are in an argument, in a verbal argument with somebody, at the moment that the person feels that they are losing that argument, the chance of a physical conflict will happen. And I think that is not that different from what happens on the world stage. And so, yeah, I mean, even if you look at Russia, and I know I'm taking a long time here, but if you think about Russia and what Russia is doing at the moment, Russia was in a very precarious economic situation. And so the idea of a greater Russia, to some extent, is that physical violence to re-establish, yeah, the greatness. And you can make the connection to the United States yourself.
And thank you to everyone. Oh, student question. Okay, let's take questions out into the reception, and you can do one-on-one with the speakers. Thank you.